Okay, so thank you very much again for the introduction, and also thank you for inviting me. As a former trainee of Trevor Pratt, I've been part of his courses in the past, so I'm really honored to be here and giving this talk. I hope I will contribute to this course. So I will talk, as Thomas said, about the introduction in ocean color. In the previous talk, yes. So Lorraine, she's been talking about, in general, about remote sensing. And she mentioned, of course, that it's the best option for gathering cyanotic measurements with sufficient temporal and spatial scale for marine ecosystem monitoring. And she mentioned about sea surface height, sea surface temperature, and ocean color. So in this lecture today, I will focus on the fundamentals and optics related to ocean color. So I will try to go from the more from the basics to the more complicated things, but it will be hopefully a good. So I will start with what's ocean color, or what we call ocean color remote sensing. And actually, the goal of using remote sensing of ocean color is to derive quantitative information on the type and concentration of the substances present in the water. And this can be retrieved from variations in the spectral form and magnitude of what we call the ocean color signal. I'll go more in detail about this, but this is mainly the definition. So basically what we see is that the water can have different colors. This probably you have been seeing that. We can see very blue, very brown, and green waters. So basically the question here is, what is the color of the water and what does this depend on? So going to the first question, so what's the color of the water? So basically what we perceive of the color depends on the intensity of the light that is reflected by the water body at different wavelengths in the visible region of the electromagnetic field spectrum. So basically what we have the white light, which is the light that comes from the sun. The visible part of the spectrum is between what we call between 400 and 700 nanometers. So that's the visible part. So that's what we see. So if we put the blue water, so the intensity of the light coming from, this is just an example now, it's coming from the water, which is blue, it will have a peak around the 400, which is the blue part of the spectrum. While if the water is more greenish, you will have a peak. This is theoretical, means not a true spectra, but will have a peak or more intense reflectance in the green part of the spectrum. So, and now going to what the color depends on, why the color changes. So basically, the color will result from the change in the type of energy, the radiation that comes from the water after interacting with the substance present in the water. So basically, it will depend on what is in the water. So going a bit of what can happen to light when it enters a body, interacts with material, let's say. So basically, light can be either reflected, so it doesn't really interact with the water body or the body, just specular reflected. It can be transmitted. It passes from one medium to another. It can be scattered, which means the change in the direction of the light, and also can be absorbed. So it is transformed. And you can see another way of looking at it in the right. You can see from the incident light interacting with a particle. So you can see the change of direction, which is all the scatter in arrows. And also we can divide it in what we call back scatter light. So what is scattered in the backward direction, so where the incident light comes from, and also in the forward scatter light or forward scattering, which is the part of the light that is scattered in the direction opposite to where the light comes from. And also the light can be absorbed. And then we get what is unscattered light that goes through the particle. So going specifically into the water, what happens? So we have incident light that comes from the sun, what we can call downwelling irradiance, type of radiance, energy basically. Part of it can be reflected. So reflected, in this case, what we call sunglint, which is a specular reflection. It's like when we look at the water and you see this, it's like looking at the mirror. You see this mirror, you see it's actually the light that is specularly reflected. So it doesn't interact with the water, but only with the surface. Another part can be transmitted, so passes the surface and gets into the water. Inside the water, as I mentioned, it can be absorbed and scattered by the different substances present in the water. Part of the scattering I mentioned can be mentioned like backscattering. Part will be backscattering in the direction of the incoming light. Fluorescence is another type of scattering. Inelastic, we change not the direction, but the wavelength of the scattered light. And so finally, after all these interactions, part of the light will emerge from the surface, what we call water leaving radiance. So basically, what we usually talk about, and when I mention what is color, I talk about reflectance. And actually, reflectance is the relation exactly between what came, what arrived to the surface, and finally what was leaving. So it's the amount of energy that is actually reflected. So put it in with the nomenclature I mentioned about is the water leaving radiance over the downwelling irradiance. And that's what we usually call remote sensing reflectance, which is actually what it's one of the products of what we can obtain from remote sensing. And this can be, as I mentioned here, related to backscattering absorption of the total backscattering and total absorption that would happen in the water. So this is, so basically, as I mentioned before, the color actually is the spectral variation of the reflectance at the water surface and the spectral variation of this reflectance. So just as an example, simple example to see, to understand this concept, we can talk about water. So the main part of the water body is water in itself. So basically this is the absorption, how it looks, the absorption of water in the visible region. So we see very low absorption in the blue and increasing towards the red part of the spectrum. In turn, the backscattering of the water have this shape. So its maximum is higher at the blue wavelengths, around 400, and decreases towards the red and more in the infrared. So if we have this approximation of the reflectance being the backscattering over absorption, we can see the reflectance of the blue of the water, just water, have a peak in the blue because the relation between absorption and backscattering at a given wavelength. So this is just an example of this relation and a simple case, which is the water. So, okay, we know how the water reflectance looks like, just pure water, but we see actually what we want to see is the change in its color. So because there are other constituents that change the color, that's why we call them optically active substances in the water, because they have this capability of changing the color of the water in the visible part. So for simplicity, in general, the constituents of the water body can be divided in four. One we call water, we have seen it. Here's like a sketch, schematic view. The other part we call phytoplankton, which actually is pigmented microscopic algae, so mainly having chlorophyll A, which is a pigment present in all the plants in land and water. Also, what we call suspended material particles or non-algal particles, because these are particles suspended in the water. phytoplankton too, they're particles. But in this case, we separate if they have pigments or not pigments. So what we call non-algal particle is actually particles that don't have this, are not pigmented, basically. And the fourth one is what we call color dissolved organic matter, or CDOM. In the literature, you will find other names, yellow substance, gelbstoff, gilving, but basically is a difference to the other part is actually dissolved. It's not particle because of the size. Arbitrarily, it's defined as dissolved and not particle suspended sediments. So here's some definition that it's very useful to have in mind, and when we talk, is what we call about the inherent optical properties against the apparent optical properties. So basically what I've been talking about, absorption, scattering, they're both inherent optical properties. And the characteristic is that they only depend on the type and concentration of the substance. Here, well, I added the attenuation, which actually is the sum of the absorption plus the scattering. That's what, so how the light is attenuated is the sum of how much is absorbed plus how much is scattered. So this is, in turn, the apparent optical properties actually depends not only on the type and concentration of the substance, but only on the illumination geometry. So something external at the substances that are present in the water. Example of this is radiance, reflectance, what I've been talking about before, because it depends, as you can see, it depends on the angles of viewing and also the wavelengths. Reflectance, which I shall mention later, before, sorry, and for example, the diffuse attenuation for the Weleny radiance, KD, is how the light is attenuated with the So, as I mentioned before, reflectance can be expressed as a function of these IOPs. Now we know it's absorption and backscattering. So basically, it can be expressed as a relations between the total backscatter of the water over the absorption plus the total backscattering. So the interesting thing that these IOPs have these properties of what we call additivity, that it means that the total inherent optical properties can be calculated or estimated as the sum of the IOPs of each of the components present in the waters. What it means, so total absorption can be expressed as the absorption of the water plus the absorption of the phytoplankton of the four groups I mentioned before, the non-algal particles, and absorption of CDOM. And the same for the backscattering, the other IOP I've been talking about. It can be the total backscattering can be expressed as the backscatter of the water, the phytoplankton, and the non-algal particle. By definition, and the size of what can be defined as CDOM, it's assumed that it doesn't scatter the CDOM. So it will not appear here in the total backscattering. So finally, reflectance in this way can be expressed as the sum of all the components, backscattering and absorption in this way. So actually I will go, so what we want or what we see, what will tell us the color, it will the variation of this reflectance, which will depend on all of these components. So I will go through the four components, water, phytoplankton, non-algal particles, and CDOM, and looking at what are the optical, the absorption and backscattering, how they look like, the spectra that will give us the result of the reflectance. So we start with the water optical properties. I more or less show you before, but I will go a bit more in detail. So basically, the absorption of the water body is generally considered invariant. So it's tabulated. You can find it in different references. So basically, it dominates in the red and infrared, and minimum in the blue. Here the spectra is quite wide because it comes from 100 to 10,000, and the visible is only this part. So if we look into more detail in the visible part, has the shape I showed you before, very low or minimum in the blue, increasing toward the red. And it's a part of this interesting characteristic of the absorption. It acts like a filter with depth. So following this spectra, we can see the red and infrared light is really highly absorbed. So it's mainly absorbed in the first millimeters of the water. And then the absorption decreases. So, and being minimum in the blue, it means it will reach deeper in the water column, and then starts to increase in the violet and UV, which is not shown here, but it wasn't here in the previous. It started increasing in the UV. Backscattering the water itself, only water. So it's really low, especially in the green and red part of the spectra. It varies with lambda to -4, around -4. And the other particular thing is that it's symmetric respect to 90 degrees. So this means lights coming this way, interacts with with water molecule, let's say. So the light is scattered in all this direction in a symmetrical way. So basically can be expressed that backscattering of the water is half the total scattering of the water, which for some things simplifies a lot of the equations and so on. Next, the phytoplankton. So the absorption, I will talk about the absorption and backscattering of phytoplankton. Basically, what we call phytoplankton absorption actually is the sum of the absorption of all the pigments that are in the cells, in the different species, whatever you have in the water body at this time. So you can see in this plot that actually you have this is the absorption and wavelengths in the X. And you see that for different pigments, here you have chlorophyll A, which I put it in green, chlorophyll C, chlorophyll B. So you can see actually there are different pigments that have different spectral characteristics. And the sum of all the pigments, it gives this dark, bold black line. So basically what we see is that there's a peak, main peak in the blue, and another peak in the red of the total. But actually, if you see the chlorophyll A is the main absorber in the blue. So this peak generally coincides with the chlorophyll A peak in the blue, and a second, the peak in the red. But just to show you that this is highly variables and depends on the species. So species can have different pigment compositions. And here you can see different colors, different species groups, let's say, not species, sorry. But actually the common thing to all of them, you can see, so these are the absorption of the group, the total absorption. So it's not just one pigment, but it's for each of the group is the sum of the absorption of all the pigments each of this group has. So basically, but what I want to show is the high variability between them. But actually what they have in common is that all have the peak in the blue and a second peak in the red. And that's what is generally useful, or it's used for determination of chlorophyll concentration using remote sensing. Okay, in general is the main absorbing constituent in the more open ocean or what we call oceanic waters. While in the more coastal areas, usually you have other like sediments that also contribute to the total absorption. Okay, so the backscattering properties of phytoplankton also is the main substance that scatters in this open ocean, as I mentioned. And usually it's modeled as an exponential function respect to wavelength. So this exponential form, backscattering wavelengths. And also it has been modeled as a function of chlorophyll concentration. So how much is scattered, it will depend on the amount of chlorophyll A pigment. That's the idea of how to estimate or be able to model the backscattering coefficient of phytoplankton. The third group is the suspended material or non-algal particles. So these are particles that don't have pigment, or what we saw. And in general, it's mainly inorganic, so sediments, which reflects more or less the geologic or the earth that is adjacent to the basin you're looking at. But also it contains its part, it has an organic part, sorry. So basically, it's usually generally assumed to have biological origin, which means part of animal, the phytoplankton fragments, mainly organic bodies or dead organisms, fecal material. All this goes through what we call non-algal particle or suspended material particles, basically. So the absorption characteristic of the non-algal particle is that the absorption is high in the blue and decreases exponentially towards the longer wavelengths. And usually it's modeled in this way. So this is the exponential, where the S here is the slope. And in different references or studies, this slope have shown to have some quite a variability ranging 0.06 to 0.13. So this is how it looks. And the difference, you can see this absorption, it doesn't have the peak, which is characteristic of the pigmented particles. In turn, backscattering also can be fitted to an exponential function, and the exponent also has shown to be variable. And actually, this depends on the size of the particles. So it has shown this exponent vary between lambda to -3 to lambda to -0.8. So it's quite variable, that can be fitted using this model. And we go for the fourth component, which we mentioned it was the color dissolved organic matter, or CDOM. And what is it, the CDOM? Okay, CDOM is a complex of mix of organic compounds of humic and fulvic acids that result from terrestrial and aquatic vegetation. So if you see when you water plant, I don't know if you ever seen in water plant and you see the water coming after passing through the earth, and you see this yellowish color of the water, that's basically also what we call yellow substance, because it's mainly dissolved substances that have an organic origin. And also it can be local or external, depending if in the sea, we are talking now about water bodies, if this comes from degradation of the phytoplankton cells or other organic particles, or can be external if it comes from river, for example. As I mentioned, the plants we water, when coming from the river, it will usually have sedum that come from all the basin that has been draining all the water. So in some cases, it has been found to be correlated to chlorophyll A, and in other cases to salinity. For example, if it comes mainly from rivers, so it depends on the place on the basin. Another interesting thing is that sedum can suffer for the degradation. And as I mentioned before, it's a dissolved substance, so it assumes that it doesn't scatter light, just absorbed, and can be the main absorber in coastal areas, which are mainly influenced by rivers, because they come from the river in itself. So the optical properties of a sedum, the absorption also is mainly in the blue and follows exponentially, the exponential function increasing toward the UV. And also with this exponential, that has been found to be quite variable too. I will not talk about scattering because I mentioned it doesn't scatter. So basically, how we go from this substance to IOP to reflectance, what the relation. I mentioned that we can separate the substances in 4 groups, like chlorophyll, CDOM, and non-algal particles. They have their own absorption and backscattering signature shapes. And following, like what we call a direct model, we can get to what would be the reflectance of this water, considering all the substances that are present in the water. Well, actually, what's remote sensing is, actually is the reverse model that we need to find. So we will have the remote sensing reflectance spectra, so that which shows the color, and we want to retrieve what's actually is in the water. So what are the substance and in what quantity, the concentration of each of them. So basically this is the job or what algorithms do. So basically the algorithm or the inverse model is what we use to estimate the physical values. So this is what I call the direct, what I've been going through with you. And the inverse models can be either empirical, so from reflectance, we can find statistical relations with the substance present in the water, with a given band or Van ratio, just an equation. Or we can go through the IOPs, or what we call the semi-analytic inversions, which actually there's some relationship, and part of them are empirical between reflectance and IOPs. And finally, is another type of product which use the multispectral fitting. So basically use all the bands, all the spectra to fit all the components and use different statistical methods like neural network and so on. So I will focus on algorithms of phytoplankton of chlorophyll concentration, which is you will see it will be mainly used for all the applications and the coming modules. So we'll focus on the standard algorithm, just a description of what it means. And so we'll went back again towards phytoplankton. So these are microscopic single cell photosynthetics. So they have this pigment that allows them to do photosynthesis, and they live in the water suspended. So like plants, so they take carbon dioxide from the atmosphere, transforming into organic compounds, and in using light energy and pigments, finally releasing oxygen. So they form the base of this of most of the food chain. So that's what they call primary producers. So why they're important and when we study them, because mainly they affect the distribution of and abundance of fishes, and they influence the cycle of elements, as I mentioned, and you can see oxygen, carbon. In this way, they influence the climate system. You will see in future modules of this series of webinars. And they contribute approximately to half of the Earth primary production. So it's very important to study. And how we study, how can we have known the amount and we can estimate. So one way I put here just pictures of different, so we can see the high diversity of cells, shapes, size. So it's really, we call phytoplankton, but actually it's really variable group, let's say. So one way of estimating them is counting cells. So cells per volume, take a volume of water and counts the number of cells. We can do it with microscopy. So looking at the microscope, flow cytometry, which just is a flow of water, which automatically counts particles in generally. We can estimate the amount as the carbon content, and also using what we call the chlorophyll concentration. So it's not the only pigment present, but since it's present in all of the groups, it can be a proxy of the amount of phytoplankton present in the water. And for this, we can use fluorometry in the lab. So using the fluorescence capability of the chlorophyll, or we can also estimate it from remote sensing, as I've been mentioning, using this ocean color, this sensor in the visible part of the region. So for doing this remote sensing, you have different type of algorithms, but in general, groups can be. One is the most used, especially in open and ocean waters, the blue-green ratio, which I will talk about. Going to more coastal or turbid waters, they don't work. You probably will see it in the later modules when they talk about coastal waters. But usually there you use other bands, the red and infrared part of the spectrum. And then you have this other type of algorithm that use all the spectrum to retrieve the chlorophyll concentration. So I will just mention and talk about the blue-green ratio, which is the most used, and mainly the standard product of all sensor is the chlorophyll concentration for open waters. So basically, this algorithm uses the optical characteristic of the chlorophyll that has a minimum in the green and a maximum in the blue. So, which I showed you before. So if we could see how the water reflectance changes when the chlorophyll concentration increases, you will see, given the absorption in the blue, the high absorption in the blue, when chlorophyll concentrations increases, the reflectance will decrease in the blue, while in the green, given it's almost constant, basically it will stay the same or slightly increase. So taking this ratio between the blue and the green, it will give difference in this slope, and it's basically related to the pigment concentration of this ratio of these two bands. And that's what the algorithm is about, has been developed fitting large amount of data collected all around the world, and by calculating the ratio and fitting this equation to the measured data. So this is reflectance measured in situ, and chlorophyll concentration also measured in situ. And these are what has the relation that has been measured, and the line is the best fit of all this data. So basically and this is done for different sensors you have maybe already heard about. So I will go a bit more on this sensor. So I talk about what's ocean color, what's the color of the water, and what does it depends on the substance present in the water. But now I go back to what we started, which is remote sensing. So what we call ocean color remote sensor. Basically, these are sensors that have bands in the visible part, because we need to see this changing in color, so in the visible, so we need bands in the visible part. Here are typical Modis Aqua, typical what we call ocean color sensor, Beers, Sentinel-3, and on the right you can see the spatial resolution of these sensors, and you can see all of them have bands in the blue, in the green, in the red, and also in the near infrared. But apart from the traditional ocean color remote sensors, we also have what actually we've been using also for ocean color is what has been actually defined for land applications. But since they also have these bands in the visible, and okay, sensors have been changing, and so now they're thought already to be useful in the water system, but they also have these bands in the blue, green, and red. And as you can see, have better spatial resolution, 10 meters, 30, 60 meters for another type of use, more coastal, useful in coastal areas. But they also have this part, the near infrared, bands in the near infrared, sorry. And more lately, it's what Prisma, for example, it's a hyperspectral. So you really have more really large amount of bands, and that's so it actually also covers the visible part and also the near infrared. And as I mentioned before, okay, so the visible bands are used, as I mentioned, to detect the change in color, basically, but we also have these near and sweep bands of all the sensor, and that's not random. I mean, they have their use. So basically, when we have a sensor, it's usually these are around 700 kilometers away from the Earth. Basically, it's detecting the information that originally the energy comes from the sun, interacts with the atmosphere, then with the water and surface, then back to the atmosphere, interact again with the atmosphere, and finally get to the sensor. So basically, we have something in between, which is the atmosphere, and which introduces kind of a noise, let's say, so something signal we don't want. And these bands are mainly used for this correction that we call atmospheric correction, so get rid of the atmosphere. So this is just to summarize what I said. So what arrives at the top of atmosphere, so the radiance that arrives to the sensor comes from what the light interacting with the atmosphere is scattered and absorbed, part is scattered and absorbed. Then part interact just with the surface, like the sun glint or the specular reflection. Part of the energy will enter the water body and will be absorbed and scattered by all the constituents, as we mentioned, and finally go back to the sensor. And also, if it's shallow, if you can see the bottom, if you can reach the bottom, sorry, the energy, the final signal arriving to the sensor will have information from the bottom. So basically, the reflectance of the water is what we are interested in. So from the total of the radiance that arrives to the sensor, the information we need or we can get information from is what's coming from the water, not the rest. So basically, the idea is the rest will be what's coming from the atmosphere and surface. So the objective of the atmospheric correction is to identify and remove the component coming from the atmosphere and the surface, to retrieve the water coming from the signal. The thing is that actually it's very low, so almost depending on the wavelength, but the contribution of the atmosphere can be really high. So the water signal is quite low. So this part of the processing is important. As you can see, if this is the radiance that arrives to the sensor, you can see there's almost no shape. So we said that we can get information what's in the water, looking at how this reflectance or radiance varies with the spectra, but we can see that actually the total radiance that arrives to the sensor has not really shape. While if we subtract what comes from the atmosphere, you can see that the clear water reflectance will look like this, so have a peak in the blue. And another case, if the water is green, you can see both the reflectance in the green, which tell us there's more green water. So basically, this correction of the atmosphere is very important to really get this information from the signal. Otherwise, we cannot see it from the top of the atmosphere. And from all these components, so the total, you can remove the atmosphere and the surface, you have to remove them. What's coming from the atmosphere can also be divided from what comes from the molecular part of the scattering and the aerosols. But the important part is actually the really molecular scattering can be calculated, it's tabulated, depends on wavelength and geometry. So that's kind of the easy part. But actually what I want to focus here is that the most variable thing is the scattering of the reflectance coming from the aerosols, which can vary in space, time, and size and type of aerosol. So basically the size of the particle. So, and what I want to mention is the typical or standard atmospheric correction, which is called which uses the black pixel correction assumption, actually. So in few words, this atmospheric correction, what it uses is the assumption that the reflectance in the near infrared is 0. As I mentioned before, one of the characteristics of the water absorption is that it's really high in the near infrared. So basically, it's really absorbed. As soon as the light arrives to the water, it's mainly absorbed. So actually, if the sensor is receiving a signal in the near infrared, that means it's not coming from the water, but from the atmosphere. So back to the previous equation, we can estimate the component of the aerosol from knowing what's coming from the water. So we have the total arriving to the surface. This can be to the sensor, This is from the surface that can be calculated. The Rayleigh that I also mentioned, it was can be also calculated. And we assume that it's really clear water, let's say. So the reflectance from the water is 0 because of this characteristic of the water that it's highly absorbing in the near infrared. So knowing this, we can estimate the absorption of the aerosols or the reflectance of the aerosols in the near infrared part, and then extrapolate it to the visible part and correct for the aerosols, knowing the shape and type of aerosols using these bands in the near infrared. So that's the basics of what we need for atmospheric correction. So basically, the ocean color reflectance, we have the radiance or reflectance that arrives to the sensor. We need to apply the atmospheric correction to get the reflectance or water radiance that I mentioned then. And then we apply biooptical algorithms. So the idea is to have this equation that gives us the concentration of this optical active substance that we're interested in estimating. So in general, this top of atmosphere radiance or reflectance is called level 1 in the images you will get, while after the atmospheric correction, this water reflectance or radiance and the concentration or different products usually are within what we call level 2. So it needs a processing, which is the atmospheric correction at least. So this can be found in data repositories, which will be a topic of the future model by Ben. So this you will get where you can get this data basically. And also to get from one level to the other one, you need to process them. And also there will be models talking about processors like Snap, CDAS, or you can do things in Python. Basically, these are processors that have been developed by the space agencies, some specifically for their own satellites, but some of them are more wide use and can be used for different sensors. And usually each space agency define the standard product for each of these level 2 data. I know Lorraine also have been talking about the level of processing, and she mentioned that it depends on the, how they call it, level 1A, level 1B, level 2. But basically level 1, it means there's no processing, just the data top of atmosphere, that's what I mean, sorry. And level 2 is already processed with kind of atmospheric correction and at the right product. So to sum up a bit about this webinar, basically what's ocean color remote sensing is the variation of the spectral form and magnitude of the water reflectance in the visible part. That's what relates to color. And basically it's used to derive quantitative information of the type and concentration of this substance that are capable of changing the water color. You cannot derive any substance you would like, but if they change the color, you might be able to estimate them. The spectral form is basically determined by the IOPs, mainly absorption and scattering of the different substances. And it's an important tool for monitoring both from the phytoplankton in the global ocean to monitor water quality, the ecosystem status for long time scales related, for example, to climate. All this will be subject also to future webinars. And I mentioned also the atmospheric correction, which actually is quite essential to get really accurate estimation, because if you have really high errors in this part of the processing, you will not be able to trust or have a good estimation of the derived products. And that's it. If you have any question, I hope it was clear enough.